So what we are going to discuss today is why iron is important. And this is actually a picture of early Earth. This is like 3.5 billion years ago. What you see here is this is ocean, which is green, which is filled with iron. And this is volcanoes, and there was no life on Earth. Then what happened is that cyanobacteria, one of the first species to appear in Earth, and it started making oxygen, and then this led to backing up of iron in ocean. Then atmosphere became iron, oxygen rich. Then other species started evolving, including human. So how do we know this happened is if you go to Australia, this is in Western Australia, this is a rock in one of those places, and this is called as banded iron formation. And this is iron oxide, magnetite, and hematite. And if you date this rock, this is like 3.5 billion years old. So this is an evidence that interaction between iron and oxygen shaped life on Earth. A lot of studies have focused on oxygen, particularly in regards to kidney disease, but very little has really looked into the role of iron. So if you take different hosts, like human, bacteria, or viruses, every one of them require iron for their survival. So free iron, on the other hand, is toxic. So what do? We need iron for survival, but the free iron is a toxic state. So then hepcidin is the central hormone which integrates diverse signals from iron stores, and then it balances between what is needed and to prevent toxicity. So essentially, when there is an acute infection, for example, in sepsis, there is an acute phase response, which causes rapid systemic hypoferemia, so the iron gets sequestered within macrophages. And so the balance between this process eventually dictates what happens after infection with the bacteria, virus, or protozoa. So naturally, since iron is critical, but it could be toxic as well, body has got multitude of transporters to keep the iron in check and in balance. So if you take a cell, there are many importers for iron, and it can enter through iron import pathways, for example, transferrin here, or through red blood cell phagocytosis, or through hemoglobin bound to certain proteins entering the cell. And within the cell, iron is stored in the form of ferritin, or it could be degraded to form what is called as labile intracellular pool of iron. But what is really important is that iron is critical for mitochondrial function. If you look at like different electron transport chain proteins, or TCA, which is a citric acid cycle, or heme synthesis, many of them are iron-related proteins. And without iron, mitochondrial function becomes defective. And as you know, many of the mitochondrial electron transport proteins are iron-sulfur proteins. And also important to note, there is only one way for iron to exit the cell, and this is ferroportin. And this is the iron export protein through which iron gets out. So hepcidin, which is the master regulator of iron homeostasis, actually regulates the amount of ferroportin which is expressed on cell surface. So this is the iron export pathway. Again, see, hepcidin is binding to ferroportin, which is a transmembrane protein on the cell surface. And if there is enough of hepcidin, then hepcidin can bind to this ferroportin to cause its internalization and degradation. So in the presence of hepcidin, the cell becomes an iron-retaining cell. If you don't have hepcidin, then ferroportin is expressed on the cell surface, and the iron leaks out from within the cell to outside the cell. So that brings us to the question, what is the role of iron in acute kidney injury? And the reason to ask that question is iron is toxic. You know Fenton reaction, where ferrous iron reacts with hydrogen peroxide, and then this leads to ferric iron and generation of oxidative radicals, including hydroxyl radical. And then there is this Haber-Weiss reaction, where there is an interaction of ferric iron with the superoxide. So because of this ability to donate electrons, iron is a very powerful oxidant, probably the most important one in biological systems. So if you're dealing with the cell injury and oxidative stress without looking into what role iron plays, the story is incomplete. So essentially, people have looked into oxidative stress in renal ischemia reperfusion injury, and this leads to mitochondrial dysfunction, membrane peroxidation, and inflammation, and so on. There are many defense mechanisms, including hemoxygenase 1, where a lot of studies have been done, NRF2, superoxide dismutase, and glutathione peroxidase. The balance between the two eventually dictates what happens after an acute kidney injury event. So these are some of the studies where people have looked into cisplatin, ischemia, gentamicin-induced AKIs, other models of AKI, and you could read these reviews about what is already known about iron and acute kidney injury. So this is one of the oldest studies by Pollard and colleagues. What they did is that they loaded iron into rats and subjected them to ischemia reperfusion injury. And what they showed is that if you inject iron and then induce ischemia reperfusion injury, there is much more AKI than mice that got treated only with vehicle. But the story is not as simple. So this is work by Jonathan Barash and others, where they've shown that iron, if delivered as a complex with the siderophore and NGAL, it actually rescues the kidney from ischemia reperfusion injury. So it is the amount of iron and in what form and when we give iron really makes the difference to what happens after acute kidney injury. So this is also a new concept which is worth noting. This is called ferroptosis. So ferroptosis is a form of iron-mediated cell death. So essentially, iron, through oxidative stress and lipid peroxidation, causes cell injury and cell death. And this work by Linkerman and colleagues, and what they've shown in this proceedings paper is that if you're able to inhibit ferroptosis by giving ferrostatin 1, then you're able to prevent acute kidney injury. So this is a summary of this ferroptosis. And again, this was discovered using this compound called erastin, which is an anti-cancer drug. And this drug induces death of cancer cells by inducing ferroptosis. And one critical molecule that's important to protect against ferroptosis is intracellular glutathione. And then it depends on this enzyme called GPX4. So now coming back to ferroportin and hepcidin, and what we have already discussed is that like ferroportin is an iron efflux protein. And if you have hepcidin, then what it leads to is a degradation of ferroportin so that iron gets to stay within the cell. And in the body, there are a few organs that really express ferroportin in high levels. For example, liver, spleen, duodenum, because this is where iron is absorbed into the body. And within liver, it's Kupffer cells and periportal hepatocytes. If you look into the spleen, it's mainly the red pulp macrophages, which are F480 high, and CD169 positive macrophages, which are near the marginal zone, are the one which are involved in iron cycling and where ferroportin is very highly expressed. So these are some of our studies where we looked into the kidney of mice under native conditions, and we stained for ferroportin to actually show that most of the ferroportin is expressed in the proximal tubules of the kidney, some in the thick ascending limb of loop of Henle, and much less in other parts of the kidney. So what are the physiological regulators of iron homeostasis and hepcidin? Under physiological setting, as you can see, there is some amount of circulating extracellular iron. But let's say if there is iron deficiency or there is accelerated erythropoiesis or hypoxia, then there is much more iron that's needed in the system. So what the body does is that under these conditions, hepcidin level drops. And this leads to export of iron through ferroportin so that there is more extracellular iron available for these processes. On the other hand, if you have a condition of iron excess, what this leads to is that hepcidin is induced. This will lead to degradation of ferroportin, so the iron gets to stay within macrophages of the liver and spleen. So this system is very tightly regulated, and it's a real-time monitoring of iron concentration and requirement in the body. So with this background, what we theorized is that ischemia reperfusion injury would disrupt renal cellular iron transport and would result in iron-mediated damage. And what we also wanted to look at is that does it only alter iron homeostasis in the kidney, or if there are changes in iron transport organs, particularly the liver and spleen. And this is the experiment we did. And in some mice, we gave hepcidin to reverse the changes in iron homeostasis to see that if there could be a therapeutic effect of hepcidin against ischemia reperfusion injury. And what we found is that after ischemia reperfusion injury, there are dramatic changes in iron homeostasis in the body. As expected, serum iron went up, kidney non-heme iron content also went up. But what was interesting and novel was that liver and spleen, they ended up losing iron. What this would suggest is that there is increase in ferroportin in liver and spleen macrophages. So essentially what this would also suggest is that if we are able to reverse this process by giving hepcidin, maybe we'll keep the iron back in liver and spleen, less will come to the kidney or serum, so we may protect against acute kidney injury. So essentially what we found is that when we gave hepcidin, these mice were protected against ischemia reperfusion injury, but there was also reversal in iron homeostasis changes as we really expected. So these are some of the creatinine data, again to show that hepcidin pre-treatment, particularly at this dose, given 24 hours before renal ischemia reperfusion injury, caused a significant protection against injury. So these are some of the histology data, again to confirm the same findings, to show that there is a reduction in injury with the hepcidin treatment compared to the mice that did not get any hepcidin. So this is lipid peroxidation, an indicator of oxidative stress in the kidney. What you see here is that after IRI, there is a lot of lipid peroxidation happening. It's a part of ferroptosis, but with hepcidin treatment, there is reduction in lipid peroxidation in the kidney. And there is reduction in inflammation based on CD45 and cells and neutrophils as expected. And as we would also expect, there was reduction in kidney ferroportin content and increase in H-ferritin, which is a protective molecule which is induced by intracellular iron. So why this finding is important is that, like, as you know, Anupam Agarwal and others have shown that H-ferritin is a very important protective molecule in acute kidney injury. If you knock off H-ferritin, then what happens is that if you subject these animals to acute kidney injury using glycerol, there is much more AKI. So not only there are changes in the kidney, but also in the spleen. Ferroportin content, which is shown by the brown staining here and by green immunofluorescence here, after IRI, it was much more. And when we gave hepcidin to these animals before, then there was a reduction in the ferroportin expression using immunohistochemistry as well as immunofluorescence staining. So the next question we wanted to ask is that if we take hepcidin deficient animals, would they get more severe acute kidney injury? So what we did is that we gave 24 minutes of ischemia, which would cause only a small amount of injury in a wild-type mice. But in the hepcidin knockout animal, there was much more acute kidney injury, suggesting that lack of hepcidin is a critical factor in AKI. And we gave hepcidin back. These animals were protected. So this is histology, again, to show that there was a very clear increase in injury in knockout animals, and hepcidin could restore protection in these knockout animals. So one more question is that what is the therapeutic implication of this finding? So I collaborated with Thomas Gans, who discovered hepcidin, and in their group, they have a molecule which is synthetic hepcidin. What it is that it's called mini hepcidin, or PR73. It has the terminal 9 amino acids of native hepcidin, and it does the same thing what hepcidin does. It can bind to ferroportin, causes downregulation, and this is being developed to treat patients with hemochromatosis who don't have hepcidin. So we got this drug, and then we injected to these mice, and again to show that even when we gave mini hepcidin to these mice, we could induce a very similar level of protection. We did one more experiment using hepcidin 22, which does not have the 3 amino acids that are required for binding to ferroportin. So what we saw is that, like, if we use hepcidin 22, there is no protection. So really suggesting that it's not only hepcidin, but the portion of hepcidin that actually binds to ferroportin and causing downregulation of ferroportin is critical for protection. So then there's other studies, and this is a group from Netherlands, which have again shown that hepcidin can reduce hemoglobin-mediated acute kidney injury, again suggesting that in more than one model, you have protection mediated by hepcidin. And hepcidin reduces intracellular labile iron, as you can see here in hepcidin-treated conditions in these cells, which are macrophages. There is much more fluorescence, suggesting that there is less labile iron within the cell. So this is a summary of the work we have seen so far, where hepcidin, if present in sufficient concentration, causes ferroportin downregulation, causes intracellular iron retention in macrophages, induces H-ferritin, which causes sequestration of free iron, and this leads to protection against iron-mediated kidney injury and AKI. So now the other question is that, is it specific to ischemia reperfusion injury? Can we show this in other models of AKI? This is a model of endotoxin-induced AKI. And what we show here is that, like, we gave LPS to these mice, and in some mice, before giving LPS, we gave Hepcidin. What we see here is that when Hepcidin was injected 24 hours before injecting LPS, there was a dramatic reduction in acute kidney injury. And in this model, it was independent of changes in kidney iron content. So these are other pieces of data to show that KIM1 and NGAL, other markers of AKI, were also reduced by Hepcidin treatment. And this is to show TNF-alpha, which is commonly used as a marker of inflammation in peripheral blood of these mice. As you can see, LPS-treated animals, there was a significant reduction in TNF-alpha with Hepcidin treatment. And there is protection of endothelium. As you can see, in LPS-treated animals, there is an effacement of endothelium in the kidney, but with Hepcidin treatment, there is preservation of endothelial fenestrae here. So these are some in vitro studies to show that Hepcidin treatment reduces serum cytotoxicity, and based on INOS positivity in the cells, and ROS generation here, again to show that and percentage of LDH release was also reduced. So essentially, Hepcidin treatment reduces LPS-mediated cytotoxicity, and the mechanisms are very similar to what we see in IRI. There is induction of H-ferritin in Hepcidin-treated animals. And as you can see here in the spleen, as Mark earlier on showed, that the presence of splenic apoptosis is a critical event in sepsis and LPS-mediated injury. In Hepcidin-treated animals, we can see that there is a dramatic reduction in splenocyte apoptosis. So in this model, Hepcidin reduces systemic inflammation, reduces kidney injury, and reduces splenocyte apoptosis. So then we went on to show that in the spleen, it reduces inflammatory signature. We looked at IL-6, IL-22, MCP1, so this is actually to show that there is a dramatic reduction in Hepcidin-treated conditions with LPS treatment. So in vitro, we could replicate the same data using a cellular model of LPS-mediated inflammation. These are macrophages, again, to show that Hepcidin pretreatment reduces inflammatory cytokine production in these cells. So, but the story is not complete. I want to show a few more slides to say that what happens after AKI. So AKI progression to CKD, does it change when we do the same intervention? So what we did is that we used the folic acid model and a UUO model to answer that question. And the way we answered that question is that we have Hepcidin deficient animals. We also generated Hepcidin heterozygous mice, which are partially deficient in Hepcidin, to ask the question, is the lack of Hepcidin worse or good for progression of AKI to CKD? So this is a slide showing pathophysiology of renal fibrosis. I won't go into this. You must have seen this many times before. And this is a slide where role of iron has been suggested in renal fibrosis. This is the singular paper where they gave desferoxamine, which is an iron chelator. And what these authors have suggested is that there is reduction in fibrosis when you gave iron chelators. But what we did is to take these three group of mice, which are wild-type mice, and Hepcidin heterozygous mice and Hepcidin knockout animals. We phenotyped them to show that there is a partial lack of Hepcidin in heterozygous mice, and the knockout Hepcidin was absent. And there is also change in systemic iron homeostasis. In the knockout mice, there is a lot of iron in the kidney, as you can see here with the Prussian blue staining. Heterozygous mice, there is no increase in kidney iron content. If you take the spleen, there is depletion of iron in heterozygous and knockout animals as expected. So then we injected folic acid, and then we followed their outcome. So what we saw was really interesting. In this model, on day 2, in HAMP heterozygous mice and HAMP knockout mice, there is less acute kidney injury, unlike what we saw with ischemia reperfusion injury. And on day 19, there was reduction in fibrosis in both HAMP heterozygous and HAMP knockout animals. And if you look at histology, again, when we do the trichrome staining with HAMP het mice, there is less fibrosis than the wild-type mice. And in HAMP knockout, again, there are more preserved renal tubules with some patchy fibrosis. And there is a reduction in renal M2 macrophages, which is shown by arginase 1 staining when the mice lacked hepcidin, either heterozygous as well as homozygous deficiency. And this is some immunostaining data to show that, again, there is reduction in macrophage infiltration with the heterozygous mice. And in knockouts also, there was some reduction in M2 macrophages. So this is quantitative data to show that there is reduction in fibrosis in both het as well as knockout animals with alpha SMA, which is a marker of fibrosis. So the next question is that if we give hepcidin back, would we be able to restore fibrosis in this hepcidin deficient animals? What we did is that we gave hepcidin back to these animals, and what we saw was that there was a dramatic increase in fibrosis when we gave hepcidin back. So this is some of the fibrosis genes, alpha SMA and TGF beta, again to show that hepcidin reconstitution makes fibrosis worse. Again, these are a couple of last slides to show the UUO model. What we see here is that this is wild-type mice, and this is heterozygous mice, which is obstructed. There is much more preserved tubules in heterozygous mice than the wild-type mice, and these are some of the quantitative data based on hydroxyproline to show that there is a reduction in fibrosis with the hepcidin deficiency. So essentially, all this data together suggested that hepcidin is pro-fibrotic, and probably not a good thing when AKI progressed to chronic kidney disease. So one last thing we asked is to give hepcidin back to the UUO model as well. Would we be able to replicate the results what we saw in the folic acid model? So what we saw here is that when we gave hepcidin back, this is some preliminary data here, that in hepcidin heterozygous mice, where there is a protection, when we gave hepcidin back, there is worsening of fibrosis. So essentially, what we see here is that there is a critical way we need to modulate hepcidin in treatment of AKI and in preventing progression of AKI to chronic kidney disease. This seems to be a very biphasic response, just like what we see with an acutely ill septic patient. There is a systemic inflammatory response syndrome followed by a compensatory anti-inflammatory response syndrome which follows. Early on, the systemic inflammatory response syndrome is what kills the patient. But later on, it's the compensatory anti-inflammatory response syndrome is what kills the patient. So we need to do opposite of that when we are approaching the patient pertaining to high homeostasis. So early on, when there is an acute kidney injury with the profound systemic inflammatory response syndrome, we need to give hepcidin to turn off that systemic inflammatory response as we saw. But then when these patients recover and move to compensatory anti-inflammatory response where fibrosis starts setting in, we need to actually lower hepcidin. So this is likely a way to really deal with this pathway in approaching and getting the best outcomes after AKI. So I'll summarize my talk here. Iron homeostasis plays a critical role in regulating cell signaling and repair responses to injury. Iron cycling macrophages play a critical role, and dynamic relationship between renal and splenic iron content dictates response to injury. Reduction in hepcidin is associated with reduction in arginase 1 positive macrophages and myofibroblast formation. Therapeutic targeting of this pathway could help in treatment of diverse acute and chronic renal diseases. Further studies are needed to establish the role of hepcidin deficiency in macrophage polarization and progression of CKD. I have many people to thank, and these are members of my lab here. And Yogesh and Eva did many of the studies, and Valentina is from Italy. She's a visiting fellow doing some of the work. Jonathan Ledesma is a fellow. And my collaborators, Marco Cusa and members of Okusa Lab, Merad Lab, and members of CIAR, my external collaborators here, and Thomas Gans, who gave me the mini hepcidin, and my studies are supported by NIH grant. Thank you.